Welcome to the final week of the NIH grant writing series. My name is Maggie Berg. I'm a research development manager at IHSI, and I coordinate the series. I'd like to begin today by recognizing and acknowledging that we are on the lands of the Peoria, Kaskaskia, Piankashaw, Waya, Miami, Mascoutin, Odawa, Sauk, Meskwaki, Kickapoo, Potawatomi, Ojibwe, and Chickasaw nations. These lands were the traditional territory of these native nations prior to their forced removal. These lands continue to carry the stories of these nations and their struggles for survival and identity. As a land grant institution, the University of Illinois has a particular responsibility to acknowledge the peoples of these lands as well as the histories of dispossession that have allowed for the growth of this institution for the past 150 years. We are also obligated to reflect on and actively address these histories and the role that this university has played in shaping them. This acknowledgement and the centering of Native peoples is a start as we move forward for the next 150 years. So with that, I'm going to launch a poll to see who is here and level of knowledge of the NIH resubmission process, as well as if anyone has actually had a grant renewed or reviewed by the NIH. Okay. Almost got everybody. I think we got just about everybody. A couple more. All right. I think I'm going to end the poll. And it looks like we got a good distribution of assistant professors and grad students today. Poor knowledge of the resubmission process, so should be a lot to learn. And it looks like only 3 people have ever had a NIH proposal reviewed. The rest are no. So great, this should be really informative for everyone today. So sharing.
I'm teaching right now. I'm teaching. Yeah.
All right, so as I said, we're going to be talking about resubmission and how to respond to reviews after you've submitted an NIH proposal. We have Say Nak and Marnie Bopart presenting today, and I'm going to turn this over to them to let them introduce themselves and give a brief summary of their experience with NIH. So Say, do you want to take over?
Sure. Hi, everybody. Good afternoon. I'm Sai Anak. I'm an associate professor at the Department of Molecular and Integrative Physiology. So I came here almost 7 years ago now, and we've been fortunate to get a couple grants successfully funded from the NIH and hope to share some of those experience with you, which I hope you find it very useful as you go through this process. So with that, I'll turn the lights on Dr. Marnie Bopat here, who's going to be my fellow presenter this afternoon.
Great, thank you, Sai. Welcome everyone. My name is Marnie Bopart, and I'm a professor in the College of Applied Health Sciences. I've been on campus for 20 years, but I've been in my position now for 14. Like Sai, I've been fortunate to have been funded by NIH, and I now serve on as a permanent member of a study section. In fact, we just finished reviewing a whole bunch of proposals Thursday and Friday. I'm still kind of recovering from that. But I'm happy to impart any knowledge that I might have on this resubmission process.
Do I just click off this poll result off, Marge, or the polling? I just click it off. Okay. All right. So we'll get started today. So today we're going to talk about one of the key aspects as you write grants. unfortunately, majority of us have to respond to the first set of reviews, and hopefully we will convert it into a successful funded grant application. We'll just dig right in. So one quick question for the audience here is that at any point, if you have a comment or a question, I don't know if you can flag or chat, put it in the chat box for Marge. I would like it to be a very interactive session so you can get the most out of it rather than just us going through slides and sharing stuff, or feel free to ask your questions. All right. So I can skip this since you're familiar with us now. Okay, so let's talk about you put in your passion, you work really hard. The first R01 is really special. I'm sure we pour our hearts out. We try to make the perfect grant as perfect as possible. We submit. And then most of the time, it's going to come back with sharp criticism. So I learned from my own PI is, again, give or take, this is my personal experience, is to read your critiques with a glass of wine by the fireplace. And the more you get upset, you just start putting the critiques into the fireplace rather than writing a really sharp email back to them. This is something I do because sometimes it's very hard for us when our work is critiqued in certain manner. But nevertheless, it's okay to overcome that and then think about it again in a second session. So usually I have like a series of reads. So the first day, usually I'm going to read it with like, oh my God, they don't like it still. But then I come back to it a week later to say, okay, what do they truly want? And then another week later, I come back and say, okay, how much of this can I successfully accomplish and make my case again to the reviewers to make it a successful grant? So I do it as a 3-step process. I'm sure everybody has their own method to this, but I highly recommend not doing it on day one because for everybody, it's really hard. Okay, so then once you've made peace with the critiques and you know, you can first, I would say also talk to your program officer. So once you get the critique, you make your point of view of where they're criticizing, talk to your program officer and gear up for preparing resubmissions. So, okay, so how do you read through the summary statement? We all get pages and pages of summary statement. As I mentioned, please discuss your critiques with your program officer. And they will be able to tell you things which are in between the lines. There have been many times where I thought X was the fatal flaw in my grant, and I was very worried about that until I talked to my program officer, and it happened to actually be Y, which was the fatal flaw, or A, which was the fatal flaw, but they mentioned X, but that was not the really sticking point that they found to be the fatal flaw. So please, I know sometimes it's kind of daunting, and we as scientists, sometimes we want to be in our little comfort zone and don't want to reach out, but I highly recommend, because program officers are there to help you because they've been there, they're listening in on the critiques, the SRO is there, so they'll be able to offer advice. And this is the key part, the second week when you're reading it, to ask yourself this question, to put yourself in the reviewer's place, to forget that you're the applicant, but then to see yourself as a reviewer and say, are these problems viable problems? Are they truly a huge issue? If they are, can you fix it? And then the other option to think about is, did it actually go to the right study section? Maybe it ended up in a study section A, it didn't have the expertise, or B, they were looking at, you might be looking at a physiological context, but they are looking only in phosphorylation and cellular signaling and mechanistic detail. So you want to think about that. Then you also want to think about, so typically, has anybody gotten grants course? We had a different expertise level and experience level in the panel. So did you get your critiques? Anybody get your critiques back? Usually you're reviewed on 5 things. your significance, your innovation, investigator,
Are you looking for an answer?
Yeah.
I had an R21. I've submitted at least a half a dozen and possibly as many as 10 trying to get an R01. I've been, except for that R21, I've been unsuccessful. I guess we're going to do that next week. But so I think the answer is yes.
So I think I'm really sorry, but all of us struggle. Okay, so hang in there. It's gonna click. Okay. So
I gave up because I could write 2 or 3 papers in the amount of time that I was spending writing grants, and I was just getting knocked down. So I'm doing something wrong.
So the important thing would be, what did your program officer say? And is it going to the right study section? That would be my 2 key questions to you in terms of, are you reaching the right reviewers?
Yes, I was.
Because the key places where all of us get beaten down is the significance and approach. Okay. So if they are convinced that this, I'm just going to throw up a molecule, let's say if it's AKT signaling, it's been abused in the insulin signaling world for a long time. There's lots of studies being done in AKT signaling. If you're just going to come back and say it's the AKT signaling again, you're really going to be dinged in the significance unless you find a brand new molecule to modulate. I mean, you need to have something which is going to be like a hook, which is going to be like, wow, this has never been thought about. Or if you find a huge gap in knowledge in the science. So for instance, I can tell a topic which we are funded on. So this molecule which we study is typically known for detoxification. Okay, so if you Google the molecule CAR, you'll find hundreds and hundreds of paper about, oh, this is known to metabolize drugs and detoxification. But the grant which we are doing is not looking at that. It's actually looking at polyploidy in the liver. How does this molecule, which is known to metabolize drugs, do control polyploid status in the liver? So I think talking to the program officer and trying to find out if it's a significance issue or if it's the model issue. Do they want you to already? So most study section want the mouse models already in hand before you can apply. So in the approach, if that is that, so I think I agree. There should be a different approach or a different place you want to try looking at with your grant. So we have one which has gone there after turn number 5 going into 6. So it happens. Every time you change according to their plan, then the panel changes and they want you to change it according to their requirements. So as long as your significance is high, I would say don't give up, because that's one thing I heard personally from my own program officer side. Still, your significance is being ranked at 3. So they find this question to be significant. So the only way I have tried to beef up my answers is by making new modern tools and approaches to kind of get me through the finish line. Marnie, do you have anything to add?
The only thing I'm thinking about are the numbers associated with the different areas for evaluation. So it's important for you not to feel upset about getting a 5 out of a scale of 1 through 9. 5 means it's a very it's a good application. And so, it used to be that you always hope to get a 1 through 3 on these different sections of evaluation. However, we're now under serious pressure by the SROs to really spread the ratings out over the scale. So we always start at a 5 with every application, and we are supposed to adjust accordingly based on the strengths and weaknesses. So the main point here is that if you get a 5 on a section, don't be upset about that. That doesn't mean that they're viewing it poorly. It means that it's average. Okay, so be able to interpret these numbers based on sort of a new scale. Okay.
All right. So yes, I also agree that triage isn't a kiss of death. This actually happened with my student. I've had colleagues where the first submission was triaged, but the second submission was successfully funded, including my own students NRSA. So again, as Marnie said, a lot of grants. So I served 2 weeks ago. So we had 85 grants, out of which 40, I mean, half of it gets discussed. So that doesn't mean the other half was not, good. At some point, we are just really trying to isolate what is the, marching down from 5. How can, I mean, people are doing tremendous amount of work. There's in excessive preliminary data. So we have to consider, okay, is this significant? Is it truly going to make a change? What is the approach? Oh my God, there's new tools. There's this exciting new avenue of research which it's going to open. So it is judged holistically. So we all try to do it. And even on the day when it's being discussed, all of us try to contribute in a sense where it's meaningful so that we can truly separate the top grant. But that, so, triage doesn't really mean a kiss of death. I would just say that in the world of relative living, unfortunately, if you go in with a bunch of grants which were really good versus, there's a lot of different factors why in that particular cycle you may have been triaged. Okay, so let's look at a score here. This is a yay score. You should all be celebrating. You get a 3rd percentile. You say hallelujah, it's going to be funded. So I don't know if you can see my score. So here you have an impact score, then you have a percentile. And then you have summary statement, of course. Here you can see this number doesn't have a A1. That means this investigator was pretty successful right away in their first submission to get an R01. And so you can glean some information. This is the program officer. This is the application number. These are the number of years the funding went through. So, and here it clearly states that currently, it was funny, my study section calls it the administrative haircut. Everybody gets a haircut. So even though you ask for a 250 modular budget, you may end up having some of these budgetary cuts. Okay. So then what are the problems which are pretty common, which are seen in a grant? So I think when I was a postdoc, I attended one of these things, and the PI who was running it said one thing which really stuck to me is that an excellent hypothesis and preliminary data can still be killed by poor writing. So even if it's an amazing, transformative grant, please make an effort to work on the writing to be clear, to be logical, and make sure that your rationale and significance comes through to potentially not a reviewer who's not super well aware of your field. So you should be able to reach a wide variety of audience, because you're going to have 3 reviewers and you need to get to all 3 reviewers and, make them feel equally excited about your grant. So I highly recommend, because I struggled initially, my first few grants writing. That was the hardest part for me, because scientific grant writing is very different from writing for journals, writing for, review articles or anything else. So it's a very different way of writing. So highly recommend taking your time to write. Of course, formatting and presentation is also important. This time, we were even criticizing some really, scientists who are very well established, leading pioneers, and they had a font size of 7, which could not be read, right? So please, I know you want to share all this exciting data, but, somebody is going to bring it up. Your presentation will be dinged. We have a lot of review panel members. So if we expect from everybody, we expect that from a NAS member or a HHMI member as well. You cannot have figures in a really small size. So I recommend that if, let's say, you're collecting data for 5 years, you end up having like 15 figures. Can you make the figures just A and B? This is not a cell paper where you need A through I at short, really scrunched formatting. Instead, show me two figures. If you're going to tell me you're curing obesity, just show me a mice in a high-fat diet with no change in body mass. I mean, that's impactful. Show your most impactful, or if you're working on a gene or and proposing a function. For us, the big figure we had was to show the ploidy counts in the liver changes with and without the molecule we are working. So just zone in on the key and show that. Then if it's a problem with writing, that's an easy fix. Honestly, you don't have to come up with a new idea. You already have the data. You just need to rephrase, make it a question that's answerable. Whether it's a positive answer or a negative answer, you still will move the field ahead. So I think that's an easy problem to have. Usually I'm pretty happy if it's just a poor writing issue. If it is, most of the time we get dinged for more experimental details. This is also this is like one of those things. If you have the tool, it's not hard to generate more data. One advice I have is we also got this is wait to resubmit your grant. You get about 37 months. If you cannot make it to the next cycle, that's okay. You can take it and push it to the following cycle, but make sure you get all the experimental details. If you have the tools, generate that data, have it, and show it. And so you can prove that. And then this I think is hard one. If they say this is not significant, sorry, I'm going in the wrong direction, then you really have to beef up the section, or you might have to bring up and say, wait a minute, I didn't talk, make this clear. Maybe there's a lot of contradictory data out there. This is kind of the norm, but look, there's a body of literature which is questioning this dogma, therefore I need to do this. So these are some of those things. Marnie, do you want to jump in and talk about? Yes.
Yeah, just real quick. One very common fixable problem that I've seen is when you try to pack too much information into a grant, too many experiments, too many hypotheses. And so many times then the investigator will come back with a leaner version, a streamlined version that then hits it. Then it's a great proposal. So just a very common mistake that is truly fixable with just a little bit of clarity.
Yes, I mean, I was also, the very first grant I wrote, one of the main comments I had received was extremely ambitious for the stage and the amount of time and the money I was asking for. This was just way too much. So
that's right. Oh, and one other quick thing too. Oh, I didn't realize you had a second slide there. One common mistake too is trying to decide if the proposal is an R21 mechanism or an R01. And so as a grant reviewing panel, we're always trying to determine, are they trying to pack too much into an R21 and it's not truly novel, so maybe it really should be an R01 or, the opposite. So just make sure that you're submitting for the appropriate mechanism, R21 versus R01.
Okay, so the other, this goes back with that experimental approach stuff. Is it feasible? So for instance, we saw it in a different study section I served. Somebody was trying to knock out 4 genes in a mice in a frame of 2 years. That's not going to happen, okay? You're not going to get tissue-specific inducible knockdown of 4 genes in less than 2 years. So again, be very mindful of the feasibility. And then this is another problem we also see all the time is the insufficient discussion of potential pitfalls and alternative approaches. So please don't be afraid of saying that this may not work, because we are all, the reviewers are also scientists. We understand that many of these times you are going to get negative data. So again, an advice I got as an early investigator was, Sai, if you spend X amount of lines on your experimental details, spend at least 3/4 the number of lines onto your potential pitfalls and alternatives. And I think all my successful grants have pretty elaborated potential pitfalls and alternatives. I would literally spell out saying, if the hepatocyte knockout doesn't show a phenotype that would say that this molecule in the liver does not do it. I mean, right? Just admit it. So I'm wrong. It just means either it's a secondary effect, I'm messing up the liver and then that's secreting something, or somebody else is compensating and that's what is going to do. And then you can say, but guess what? I have the flux allele. I can make some educated guess of what other knockouts I can make. So I'm very grateful for the advice I got in my second year. And he said, just say it. It's okay. And then when you're submitting the revision, please don't submit the same thing without any substantial changes. That's never helpful. And I always ask any or all of my colleagues, I'll just send a blanket email and saying, I know if anybody is willing to read my grant, I'm more than happy to share it and I'll do the favor back. So again, send it out. Don't be afraid to be critiqued, because if you critique locally, then you won't get that critique from outside where it matters for the funding line. But that's my approach. Marnie, do you want to add anything on this slide?
No, this is great.
Okay. All right, so these are the hard to fix problems. Again, be very mindful of, this could again route back to the study section and the institute if you're not targeting the right institute or the study section. So our Ploidy grant had some conflicts, which I didn't know when it went back to the same study section and had to be kicked out to a reproductive panel. So reproductive panel is not going to understand liver metabolism. It needs to be with the right group. So my score really tanked when it went there. So, and they didn't find it significant. So you have to really go back and assess your summary statement, again, in collaborations with your PO. I would say talk to your PO, talk to your peers, talk to colleagues who are funded from the same institute, which I have done and found it to be useful. It's very important that you can convey that there is a significance in this work, because that would really be a fatal flaw if you cannot convince that. And then if convincing needs that extra piece of data, I would not resubmit till I have that extra piece of data. That would be my advice. And we've done that before. My students, NRSA, we waited for 1 year because we wanted to do a cancer experiment, which takes 8 months to develop cancer and then show that. And so it was a triage grant to a 4 percentile because we waited that 1 year to make that case that this was important. So I highly recommend doing that. And then, so it has to be innovative, clearly. Unfortunately, this has happened with me in one of the grants, that one of my aim was kind of scooped because partially some other groups, some pieces of it, and we had already finished a lot of pieces of it. So we had to remove that and come up with a new aim, and then showed in our, BioSketch and other places that this work is actively being done and over. So you will get the positive points that, okay, they made an effort to fix the grant, the aim, which is already completed. So you have to make sure it's innovative. And then this I have not seen much, but sometimes you can propose methods which are not suitable for testing the hypothesis. So that would be a problem. I would, yeah. So you really have to think about what you're going to do with your proposal and fixing these hard problems. Okay, so contacting the program officer. So if you're just about the pay line, you can ask. Again, it depends on your relationship with your program officer. So again, everything is like apples and oranges. Everybody shares a different bond and different comfort level. I had a program officer where there was only 1-line questions and 1-line answers, and I was still able to get funding. And I have another program officer with whom we can drink a cup of coffee and chat about family and chat about everything under the sun. So it's going to depend. But mostly I have found that even if their reply seems to be curt, they want to help. And that's how I interpret it. There have been times when our program officer said, "So if you don't have that data, I recommend you really seriously think about it," because they cannot say submit or not submit, but they'll say, "Hey, are you sure that this amount of data is convincing your main point?" I usually take that with a lot of weight. So then I'll say, okay, she makes a fair point. I really have to do more experiment. And as I said, their viewpoint is important because sometimes I have gravitated in the wrong thing as a major fatal flaw, but it's not. So Marnie, do you want to add anything? Yes.
You always know. Yeah, so this is such a great point that you bring up with the program officer being a little curt when you try to contact them for feedback. That was my experience, and I was just so offended. I was just like, oh, I got this one-liner sentence. You know, I was 1 percentile over the pay line, and she just basically said, resubmit. You know, and I'm like, oh, that hurt. You know, like ruined my week. But it ended up getting funding, you know, so it was funded. So, you know, just like you said, you know, you when you do need to contact the program officer, I find it to be very helpful, but don't expect to sit over coffee every time, you know, and have a nice chat. That's not going to always happen.
Yeah. So apart from that, so my example was I worried too much at my 3rd year that my first paper as a last author was being a huge problem, and that apparently was not, but instead another data point was. So they will clear it up saying what was said in the meeting. Okay. This is again a discussion deciding whether to resubmit. I would say that consider all the points we have discussed until now when you make that decision to resubmit. I would say if your significance and your approach seem reasonable, they haven't found that, you need preliminary data, which will take 2 years to generate, then, I mean, those are the things for you to really consider and think hard on before you decide on resubmitting. Marge, do we want to open it up for discussion at this point, or?
Yeah, if anyone has questions, you can either put them in the chat or you can unmute yourself and ask your question.
I'm just going to switch on the light to my room. I'll be back. Oh, my light. It's going to be dark in the corner because
All right. It looks like no one has questions right now, so we could probably keep going.
moving then. Yay, everybody's decided to resubmit, I guess. Okay, so these are some of the numbers. If you listen to NPR, I'm all about the numbers in the evening, you can hear. So you can kind of see that from 2006, so this is until 2015, so 5 years ago. So your first submission is in red. So about 30 percentile of the people on an average will get grants in their first submission. Actually, it's first resubmission, my bad. The first original is much lower. So as I said, you're not going to get funded. You will get funded, but very small portion will be funded at your first try. But second try, you significantly went up the bar. And then we no longer have secondary submission. So NIH has taken away that program. Instead, you come back as the blue line again. So you just go like 0 and 1. That's it, like a digital system. So you can come back, resubmit as a 0. And Open Mic is a fun blog if you have not done it. I really like Mike's sense of humor. Okay. Then if you look at these scores, these are all the impact scores. And as I was just mentioning, 50 and below usually don't get discussed, but these are the numbers again. These are new grants, these are renewals. Again, if you did really well with your scoring in the first cycle, this is A0 awards, then you're going to have very well high percentage that your resubmission is probably going to come through. Even though on paper they won't say, hey, resubmission gets a special treatment, but we all appreciate how much work you do. Everybody reads that one-page resubmission summary. We see how responsive you've been to it. And usually when the second time around, the grant comes back in a much polished, like just Marnie was saying, very nicely put in, laid in a very well put way where all of us can see the significance is highlighted, preliminary data and everything. So when you come back the second time, even the new grants have a high chance, 80% chance of being funded, and so are the renewals. And I'll be honest, this time in the study section, we saw a lot of A1 renewals. So this happens. I mean, whether you're with 42nd year A1, 32-year A1s, those are happening. So everybody has to revise and resubmit. And then this is impact score, and again, A1 funding rates. So this is basically funding rates depending on their initial peer review score. So they're combining the first two figures and saying, if you had very high score to begin with, you're going to have very high success rate. It's pretty much reiterating the same thing. Okay. But again, I would say that these people will be moved here as well. They don't stay just here, whether it's A1 or A0. So some of these folks get moved into these cycles. This is more funded resubmission application. You can see that 6,000 and 3,600. So a lot of grants do eventually get funded. So funding rates for A1 over A0 score. So if you had a very good A0 score, you come in with an A1, 80% chance you're going to get it. And with the renewal, it's a 90% chance.
So we have a question in the chat.
Yeah.
They say, so then how do you interpret or deal with getting bounced around? For instance, scored reasonably the first time, triage the second time, and so on.
Okay, I have a personal experience with that. So I'll openly share my scores with you guys. 23, 30, 24, 46, triaged. Okay. You don't do anything. You just drink a lot of wine. No. But I think what happens is, unfortunately, when this happens, this just means either the panel is changing more too often. A lot of the same people are not seeing the same grants, and this was one of the facts. So when we revised and resubmitted, unfortunately, the number of grants which came in at that time, it's a relative comparison. Even though my score was the same, the overall score dropped. Individual sections improved, but my overall score dropped because everybody else who came in with my pile did much better. Okay, unfortunately, we live in the world of relativity. The third time around, we changed everything what the reviewers wanted, right? Which would have been now an A0. But my study section just had a huge turnaround. So 50% of the members retired, bringing in new people, and they hated the new version. They wanted A0 of my original A0. They're like, why is I doing the same old stuff, which nobody, so it's very unfortunate. Then we took, then I did that, I was coming at the fourth year, right? I was getting desperate to get a grant. So we put the polyploidy grant, which was, at the 16th percentile, was not going to be funded. I had just come off the ESI status 2 months earlier. They said it was not going to be funded. 13 was the cutoff. So I had to resubmit. So we put some effort on that because I knew 16 percentile is a pretty good score not to not resubmit. But then we put an effort on this one as well, thinking that my whole department thought the other grant, which is getting bounced around, is a much better grant. So we put in a lot of effort on that and ended up with a triage or 46 or something like that score, which I said. But my other grant came through at the 4th percentile. So I was about to give up on that grant, thinking, anyway, we are doing that. My student gets an RSA on that. So I went to the NIH new investigator. They had a workshop to make sure we remain continually funded. We are able to bring in our renewal. And I was talking to somebody exactly the same question. What do I do with a bouncy ball? And so, and the answer was to, hang in there, go back, reread everything. So we took 2 years. So we submitted this June or something. It's going to be discussed or not discussed the following week. So it's 22nd and the 23rd. So it's coming up. So I'm really hoping. I talked to my program officer, and she always says that they're super excited about the significance. It's something really novel, but they want more and more preliminary data. So one caveat is the protein which we are trying to express. Again, mice are smart. This is a huge scaffold protein. We've tried to make transgenic mice to express this. We got 10 positive lines, but the protein never gets made. You have the gene in, but you're not getting the protein made. So the only way to do it would be to force feed this protein by a viral infection system. And then so at some point they were stuck why I was not showing the transgenic data, because it was negative. And then I talked to my program officer and said, why don't you just say that this is negative? The only way to do it would be to do the virus. So that's what we've done, is to just go ahead and admit that, because I was very worried that'll be a fatal flaw, that they're going to come back and say, look, I can't even make this transgenic. But we had 10 mice lines in different regions integrated in it, but won't express the protein. And I'm hoping that's going to pay off. We'll see. But the other 2 aims, they don't seem to ding. They said, oh, this aim you've already published, your grad student published it. So we just changed it with a new aim, which anyway we were doing. And let's hope. So if you're committed to the project, and if you're passionate about the project, keep going. We floated the project with campus board awards. That's how I've gotten it, or try to get my students or smaller funding to kind of keep it going till you get the big R01. But I would say that if you personally are not convinced, then that's a time to kind of reevaluate. I'll let Marnie weigh in on it.
Oh, you did a great job answering. In terms of, I'll just add really quickly that I was always told submission of grants is a bit of luck too. You don't know who's going to receive it and review it. And sometimes you get a reviewer that's, very loud, their voice carries pretty heavily, and there's just nothing you can do about that. And so, like you said, just hang in there. And if you really like this project and you're committed to it, you really need to just stick with it and continue to try to submit.
Okay, as you know that the resubmission is valid for 37 months, but your ESI status doesn't. So I tried to ask them when I knew I was going to cut short into that, that 16 percentile, but they didn't extend my ESI status. So again, you can get an extension in case you had a baby at a certain time. I mean, there's few or a medical emergency. There are some things. So look into the NIH website, email them in advance if you are eligible for an ESI extension. I know my colleague who is a neonatal ICU internist, he was able to get almost 7 years of his training waived after his MD degree because he was training. He was not being a faculty. So those are all the things. So call in and find out if your ESI status is valid. And all the assistant professor, I highly recommend using the ESI status. My husband, who's also a faculty here, really benefited. He got a 10 percentile boost because of the ESI status. If you can use it, just use it. It's a lot of percentile not to try. So, and then, yeah, we already talked about does anybody want to talk about the 1-page introduction? Is everybody familiar with that? I think that's the only new thing.
I think I was going to cover that next.
Okay. All right. So I will hand the baton over, I guess. These are the dates. So you can see you get an extra month off for the resubmissions, which is nice. I don't know. It makes me feel special. They are all worrying about February while I have to worry about the March deadline. And then I'll let Marnie take over.
Margarita's
We'll
on.
stop the share.
Okay. Okay, 1 second. I've lost the new version of the, here we go. I think that's it. There it is. Too many files open today. Okay. I think Margarita, I saw something come up in the chat.
I think it's a open mic blog, I think.
No, Margarita, Tehran. There is something there.
I wanted to share, I wanted to just to share that sometimes this type of information, and I really appreciate your presentation. It's that some of this information is not always shared with all faculty, especially what you were saying, Sanji, about early state status and how many times you can apply. I think this is very important, and even underrepresented minorities or minorities in transition of careers get this type of information. I have to tell you up front, I was not aware of any of this when I arrived to the campus. And then you learn it kind of a little bit too little too late. And you think this is important for the type of mentorship that we give to our young faculty and young postdocs. I think I want to make it a very important point for sharing.
That's a really good point, Margarita. It's a great resource no matter what stage you're at. I would agree. So a lot of this information I did not acquire until I started reviewing actually for NIH a little late. And so it was a pretty painful process, I would say, submitting until I had a better sense of what's the expectation, and I didn't realize the expectation was so high, until I started looking at some of these samples that I was actually reviewing now. And I was like, wow, this is amazing science. And this is an amazing proposal. And I'm like, I really have to pick up the pace here. I really need to do much better. So yeah. Okay, so this will be really quick. I just wanted to comment on writing the introduction. So you've decided you're going to resubmit your application. And there is a trick, I think, to writing this one-page introduction. And so here's what you're going to include in the introduction. Of course, there's going to be a summary of what changes you made. You're certainly going to respond to all of those comments. And you can politely disagree with a reviewer's comment if you have a good justification for that. The introduction is 1 page or less in length. And yeah, it's hard, honestly. It's challenging to be able to comment on all of the reviewers' critiques in 1 page or less. And so here's a couple of examples. And what I wanted to point out is the very first paragraph. This is the trick. So that first paragraph, you want to make sure that the reviewers are aware of the positives that came out of the first review. So here it was this proposal. This was not mine, but another sample that was given to me. Where the proposal received 21st percentile. That's going to strike the reviewers as, wow, this was a strong application to begin with. And if you don't state that up front, they're not going to know because, as a reviewer, we do receive the summary statement and I do read them, but I can tell you most reviewers won't take the time to go through all of those comments. They're just simply going to read this page. And so, I like how they bold-faced some of this information here, how you've added extensive new preliminary data and you've developed new reagents and models. Whatever is the main sort of change that has been made should really be highlighted in that first paragraph. That's just so incredibly important. And then here's another sample too, and it's not bold-faced, but you can really try to bring out again some of the positives. Excellent track record. They mentioned there are no major weaknesses. The study is considered cutting edge scientifically. Like those really, really highlights. Those highlights should be placed right there. Now, I think the challenge is really trying to not address each critique point by point like you would with a paper. So, when you submit a manuscript and they give you comments, typically you will reply back to every single question comment. You don't have the room here because you're replying back to basically, 3 reviewers and you've got to do it in 1 page. So you need to be able to condense some of the thoughts. So this is where it takes you some time, I think, in reading and rereading the comments that you get back and realizing that, okay, reviewers 1, 2, and 3 all had the same comment. Okay, so I'm going to then address those reviewers at the same time. So if you'll see here, point 1, development of AIM-3, all reviewers expressed significant enthusiasm, but they all had, some type of critique. And so I'm going to try to address them all at the same time. And so this does take some time, I think, to effectively, put together a nice summary of your response and in a very strong and clear manner so they understand. Don't underestimate the significance of this introduction because we're looking at it, as part of the review process, we have to write a small paragraph on whether or not you address the main concerns of the first review. So there's a special section for that. So here, if you decide, in the case that you want to request the same study section, you need to sorry, let me just reread this. You're going to, I wrote here the reason why I decided to submit to the same study section because it said that overall the review panel considered the application to have merit and the weaknesses which the panel members viewed as correctable. Okay, so this kind of goes back, this goes back to what Saeed already had mentioned, looking to see whether or not you want to resubmit based on their comments. I'm just going to go ahead here. So this is the last slide for the presentation, and here are just some last minute tips. So you need to, after you have, again, reviewed all the summary comments and you had your wine, you need to revise your research plan. So you're not just going to simply comment back to the reviewers, but you certainly are going to make the changes. You're going to throw out or revise the weaknesses that were identified by the reviewers. You're certainly going to add new findings and your own improvements. And so they may not have picked up on something, but you have collected new data since your submission, and you might want to include that to make it a stronger application. Again, you're going to then respond. You're going to create that 1-page introductory statement, and I put here, however, you're not going to respond point by point. You're going to condense the information and respond back collectively. And then you can disagree, as mentioned, but you want to be very respectful with your disagreement, and it has to be scientifically based. And then I think it's really important to find samples of introductions online or from a colleague or a mentor. I know for me, I looked, I asked many of my colleagues, and I got 3 different samples, and I was able to then formulate my own response based on that sort of structure. And then finally, as Saeed already mentioned, you may not want to rush in resubmitting. You may not want to rush in putting this response together. You might want to wait, not you don't want to resubmit that next cycle, but maybe wait till the next one. However, I will tell you that I have been co-PI or co-I on different proposals where my colleague wanted to resubmit right away. And I was like, no, we need to collect this extra data, you know, and they did it. I was like, fine, just go ahead. And they got funded, you know, and I was like, you're kidding me. I mean, and that had it been me and I had waited, I would have lost the opportunity. But, you know, and this has happened twice, I would say, that they just like immediately jumped on it and got it resubmitted right away for the next cycle and didn't want to wait. So you know, you sort of take your chances there, but maybe, you know, it's sort of, I guess, you just never know. Again, sometimes it's just luck. So that's all I had then. So any questions maybe about writing that one-page introduction or response to the reviewers? Nothing. Maggie?
Yeah, I guess you explained it really well. Yeah, Say, do you have anything to add about tips for writing that one pager, responding to reviewers' comments?
Yeah, I mean, I think Marnie did a good job of covering all the points. The only point I would add is, so this was an advice from my PI when I got the 16 percentile, just like how Marnie said, highlight it. And then, you know, my mentor said, don't use the whole page. It was a high impact 16. Just pick the major point and say, look, this was dinged because of lack of this particular expertise, which I have already now established and I have preliminary data. So my response to review on that was only 2 paragraphs, which I was very worried about when I submitted, because you feel like, oh my God, they're going to think that she's trying to not even fill. But honestly, there was no other big major weaknesses. I feel like many of us question a lot, like, oh my God, am I doing wrong? So at some point, just having that confidence and saying that, okay, this was the major weakness, I fixed it. This was a very highly scored grant with very high innovation, and that's all I got, right? So that's something which I did. So, but mostly I use the entire page because there is a lot of nuanced comments on approaches.
I wanted to make one last comment about the scoring issue, the scale. So the scale is a high of 1 versus a low of 9. And, you know, I fear that if you're submitting for your first time and you get a score of, you know, again, 5, that you're going to feel deflated and you're not going to feel good about yourself or your proposal. But you have to be careful again, because in our last review, we didn't give a single 1 to any application. Like there wasn't a single investigator that received a 1 for their final score. And so even a 3 was considered very high. All right. So when you look at these numbers and, you know, and you see these sort of average scores, it's okay to be average, you know, and shoot for your 3s and possibly 2s, and you may get funded on your 2s and 3s. But 1 is rare anymore.
So we have a question in the chat. As a reviewer, do you think that issues with impact can be argued effectively in the introduction to revision?
Impact, you mean significance?
I think that's what she means. I don't, unless she would like to clarify.
Yeah, I mean, yes, I guess some places call it impact. I guess NIH calls it significance, but is that a place to argue if the reviewers didn't find it impactful or significant based on how you argued it in your first submission, is this a place to highlight, like, this is why I think you're wrong, this is why I think it's very impactful? Or do you not address that in the introduction, just try and beef it up in your main body?
Well, I think it just depends on the situation. I don't think we have a straightforward answer for you here. I can tell you an example of where I reviewed an application recently where the data was really amazing, but they just didn't give a bigger perspective of the problem, the societal or health related problem. And I couldn't then sort of tether it to that situation. So were they talking about myocardial infarction, so, I just didn't get any sense of what was the bigger problem. And in that case, I would say, yeah, I think they could easily just come back in with a paragraph statement that, said, this is, the bigger societal problem that I'm trying to address. That's part of significance, but there are other components to significance as well. And so now we're starting to emphasize the scientific premise. So any preliminary data or backing of the reason why your hypothesis is likely to be successful is underneath significance. So I think when we hear significance, it's always, again, societal problem that we're trying to address, but that's not it anymore. Now, what we're told to address for our score is the background and the premise and sort of the backbone of the grant. Like, what do you know already that makes it so that it's likely that this will be successful? So I think that's why this is a hard question to address.
As Marnie said, we do have a component called prior rigor to this, research. So rigor has now become a huge part of this significance, like how much supporting evidence, not just from your lab, but from different labs, you can come to address this problem as. And specifically, if you think that the reviewers understood it wrong, we did this for my RO3 to say, they said the PI doesn't even understand what she's trying to do. So we had to come back and respond saying, no, we did. It's just the choice word which was used in the background and significance, which was inappropriate and gave that impression that this problem is not significant. So for instance, we work on cholestatic liver diseases, and it's just many liver diseases end up having cholestasis. So it was not put in the appropriate way. So they thought that this, as a new PI, I didn't know what I was doing. So you can write it again. You have to stick to the facts, keep to the science, and say, I was apologetic and I said that was not the right choice of word I should have used, but instead to highlight the impact, we should have, stated some of this literature and then bring the significance in place. And so our revised version was funded. So I think you can. So if you think that was a major hitch and your program officer says, okay, this is where they got stuck, I would say definitely put it in the front, and then also do it in your significance area. Maybe you have to reword the entire significance section to kind of highlight that it's now significantly changed to showcase whatever you want to highlight right now, which was lost initially because of poor grantsmanship or the way things were phrased or the logical flow was different. So yeah, it's no harm to try. I'm always a person that if you don't bat, you're never going to get a hit.
Does anyone else have any questions? I had one question more on the writing portion of submitting a resubmission. So how do you typically signal that you've changed things within the text outside of that one page?
Ooh, that's a really good question. I've seen that done a variety of different ways quite effectively. So some people will write the revised version in like red, some of the areas, or they'll underline. So I typically use a red font, which is probably not good considering some people can't read red, but that way they could easily see those sections that have been altered or changed. I don't know about Saeed.
I just use a line on the side and then just say the left indent shows a bold line, because sometimes, one of the mistakes as a new investigator you also make is bolding too many things, bolding only important things, or italicizing only the important things is the key, because this cycle we had 3 grants in which every line was bolded. It's not convenient for the reviewer to read every single line which is bolded. So I have usually used a single line, but I have seen a lot of blues. So these days I've seen a lot of blues to show the change, where the change is. And typically, if I just change an entire aim, I just take a line to the margin and mark the entire thing rather than the whole section looking a separate color. So I try to do that. Yeah. Anything to make the reviewers read it easily. So this is an acid test, which I ask my students to do. I'll send them my grant before submission, and they have to read after they're very tired, almost at 11 when they're hitting the bed. And if they still understood my specific aims page, then I guess it's time to let go. If not, and I'll do the same too. On a Friday night, you have a nice meal, you enjoy your evening. Just before hitting the bed, just take that one page and then see, despite all the distraction and exhaustion, is it still keep your interest? Mm-hmm. If it can't keep your own interest, then it just needs to be reworded.
Mm-hmm. That's a really good point. I would say, as a reviewer, if I can't understand the specific aims page, I'm sort of done. I'm angry. It makes me so upset when I don't get it the first time. If I have to work really hard to understand the point of the grant, then I get upset, and it comes out, I think, in the review.
All right, do we have any other questions? I'm going to quickly share this last slide. Okay. All right. I think we might be wrapping up if there's no other questions or discussion. Thank you very much to Say and Marnie both for presenting today. I think they had a lot of really great advice, and hopefully all those people who marked poor on their understanding of the resubmission process are more up in that higher knowledge area now. So this is the last session for the seminar series. There is a peer review workshop, but that is only for our selected group of nominees. If you in the future would like to be included in that cohort, you can contact me and learn more about how you can be nominated. We do accept self-nominations and as well as nominations from your department head. So we are not going to be offering the series again until fall 2022. So we'll actually be skipping next fall. So you can look for a call for nominations in spring of 2022. So proceeding that fall. In the meantime, if you ever have any questions or need assistance regarding proposal development, or finding collaborators or resources, you can always feel free to contact me or any member of our research development team at IHSI. So I hope that everyone has a great rest of their semester, and good luck on all of your grant submissions.